All right, this video is going to cover visual sensation, I should say. So we're going to spend a little bit of time talking about the eyes, how they manipulate light, and of course how they transduce that light into electrochemical signaling. We'll very briefly outline the visual pathways that then lead to perception, but for the most part we're going to hang out in the eyes, and I guarantee you there's plenty of action there. Okay, here's the required reading for all this stuff to make sense. Okay, so let's review a bit of eye anatomy here. So there's a lot of things outside the eye and inside the eye that are going to support the function of the retina. And the retina is where the magic happens. That's where we take light and turn that into electrochemical signaling that eventually leads to our visual perceptions. Okay, so just a few accessory structures here. So first of all, if we're going to put the eye somewhere, there has to be a space, and that's the orbit. That's just that hole in the skull there where the eye is going to sit. There's a cushion, of course, the orbital fat there, right? We don't want to put eye on bone, so we have a little fat cushion that it sits in. On the outside, we all are familiar with eyebrows. These help show our facial expressions, and they keep perspiration out of our eyes, and as long as you can see them, if they're darkly colored, they also prevent some glare from going into the eyes. The eyelids, of course, are there to provide a little physical protection and help put tears down and moisten the eye. The eyelashes are what we call guard hairs. That is, they are these kind of long hairs that form a filter and hopefully keep junk from floating into our eye. The conjunctiva is a mucous membrane on the inner eyelid right over here, and then on the anterior or front of the eye there. The whole purpose of this is to moisten the eye, and when it becomes inflamed, make the eye nice and red there. Then we have the lacrimal system. As the name suggests, this produces tears. Tears are, of course, wet, so they moisten the eye. They also have some nutrients and some antibacterial enzymes to help keep the eye clean and well fed. Here's our good old lacrimal system here. So we'll create some tears. They'll come on down, right? And they should flow on over down through the nasal passage there. So there's this kind of constant flow of tears over our eyes. The eye is moved around by a set of 6 extrinsic muscles. We've got 4 rectus muscles right here. Rectus muscles have straight attachments, whereas the oblique muscles, there's a pair of those, superior and inferior, have these perpendicular attachments, and that causes them to be a little bit strange. So if we look at our superior rectus here, hitting the top of the eyeball, and the superior oblique hitting at a completely different angle, think about what happens when that superior oblique contracts. It's going to pull the eye in this direction. In other words, it's going to pull it the top down and actually make us look down, even though it's on the top of the eye. On the other hand, when the superior rectus contracts, what's it going to do? It's going to pull the top of the eye back and make us look up. So the rectus muscles will do pretty much what their name says, and the oblique muscles do pretty much the opposite of what their name says. Now, because we're not quite hitting straight on here, we're kind of at an angle for the rectus, as well as, of course, the oblique muscles. That means there's going to be some rotation of the eye. That's the torsion there. This is a really nifty table if you're into eyes and their movements. I'm not going to focus on it too much in this lecture. I do talk more about this in another video, but not too critical for this class. Here's a little summary that I made for you. So if you're looking straight ahead, let's just say there's even tone in all of my extrinsic eye muscles there. If I want to look toward the eye, the medial rectus helps my eye look medially. If I want to look away from the eye, over here, this is the nose, by the way, The lateral rectus moves my eye in the lateral direction. Now, while I'm over here, the superior rectus will help move my eye up, and the inferior rectus will help move my eye down. At midline, it's a combination of the superior rectus and the inferior oblique that move me upward. Remember, the obliques, they're always doing the opposite of what the name says. And to look downward, it's exactly what you'd expect. When I am adducted, that is the medial rectus is active, now it's mostly the inferior oblique that helps me look upward and the superior oblique that helps me look downward. The rectus there is just going to cause twisting of the eye. Okay, what do we care about here? Well, the obliques do the opposite of what their name says. And which cranial nerve controls them? Luckily, you only need to know two of these. There's the lateral rectus, of course, controlled by cranial nerve 6. And then there's the superior oblique, controlled by cranial nerve 4. Everything else is cranial nerve 3. So that's kind of nice. Now, if we dive through the anatomy here, we're going to start on the outside. So we'll think about the tunics. We're going to go from outside to in. So the first tunic, the outer fibrous layer there, we have our sclera, the white, and the cornea, which is clear. This is a modified form of the sclera that's transparent. The sclera is very dense connective tissue. It's very tough, gives the eye good protective structure there. The cornea is transparent, has some stem cells in case it gets damaged. It can regenerate, but its purpose is to be fairly rigid and transparent. That way light can come through. Moving down a layer, we have our choroid layer, shown right here. So between the white and the retina there. When you hear choroid, you should think vascularization. So this is where we have a whole bunch of blood vessels, and this helps keep everything well fed and, of course, remove wastes. Also in the middle vascular layer, we have the ciliary bodies here. This is actually a ring of smooth muscle. It's just cut off in the illustration, so this continues around and forms a ring. This is what's going to control the iris. The lens. The iris itself is smooth muscle, so the ciliary bodies will control the lens, focusing the light. The iris is another smooth muscle. Again, it's cut off. It forms a ring, just like the ciliary bodies. And in the middle is that hole that is the pupil. So depending on how constricted the iris is, that will determine how big the pupil is and thus how much light comes in. Finally, we have our inner layer, and that's the retina. This is where the action is going to happen. This is where we have our sensory cells and our nerve cells. There's no vasculature within the retina, but there is vasculature, of course, surrounding it on both sides there. Okay, now we could think not just in terms of layers. Let's think about the optical components. Okay, so if I have some light here, we turn on our light bulb. That light is, of course, first going to pass through the cornea there, which I've covered up with the ink. Remember, this is transparent, it's not white, so it's a modified sclera. And the whole point is to let light come into this anterior chamber. Now, in the anterior chamber, we're going to have our aqueous humor that's created back here in the posterior chamber. So anterior is in front of the iris, posterior is behind it. The aqueous humor is created by the ciliary bodies, and then this kind of watery fluid is going to flow on through the pupil and enter my anterior chamber where it's reabsorbed by those scleral sinus veins. All right, so our light has now passed through the cornea, through that aqueous humor there, and then now we're hitting the lens. This is going to focus the light here. Now remember, these ciliary bodies are a ring-like structure here, and connecting them to the lens are these suspensory ligaments. Depending on how contracted the ciliary body is, that will determine the tension that these ligaments put onto the lens. So if my ciliary bodies are dilated, not contracted, there's a whole lot more tension on the lens and it's pulled to be thinner. However, when they contract, notice what's going to happen. They're going to move kind of toward the center. Their inner diameter is going to decrease, and there'll be less tension on the lens there. That is going to change the plane of focus a whole lot more on that later. Okay, so we've passed through the lens, that focused us, and then we hit this vitreous body here. That's this big old transparent jelly right here. And that is just to make sure the eye keeps a nice round shape, whether it's sitting still or moving. There's this little hyaloid canal here present in the vitreous body. There used to be an artery here feeding the cells that made the lens. We don't need that anymore, so it's gone. But we can see remnants of it running through that vitreous body. The whole purpose here is to not absorb a bunch of light, but to be fairly supportive of eye shape. So keep the eye nice and round. Then, of course, we hit the retina, and this is where the magic happens. A couple of important areas, the least important of which is that anterior margin, the ora serrata up here. Everything in front of this is no longer retina. Everything behind is retina. Now, what do we care about? Well, there's the periphery. As we'll discuss later, this has a whole bunch of rods in it. And then, of course, there's the fovea, where we find most of our cones. This is the center of your visual field. If you're looking at this dot right now, it's in your fovea. Nearby, we have what's called the optic disc, and this is a blind spot in our retina because it contains no photoreceptors. It contains the axons that form the optic nerve and the blood vessels that are going to enter into the eye. So, since we're occupied by axons and blood vessels, we can't put photoreceptors. And therefore, this part of the retina sees absolutely nothing. So each of our eyes has a blind spot. Of course, we don't see it unless we go through great lengths to make it visible to us. Our brain just lies to us and paints over our blind spot. Okay, let's see if you have any blind spots in your understanding. So you can scan this with your phone. I will also have a link to this first pit stop in the description of the video. So check that out. Make sure you're getting something on that. If anything else is unclear, get at me so I know. So the first thing the eye is going to have to do for vision to work properly is to make sure we're getting enough light coming through the pupil so that we can actually see things. Not too much, not too little. That's the job of the iris there. Control the size of the pupil. And then make sure that light is focused on the retina. That's the job of the lens there. So we need to manipulate our light. First of all, what in the hell is light? Is it a particle? Is it a wave? Is it both? Yeah, sure. It's all of those, depending on who you ask. So light is made of photons, little particles that act like waves. Ooh, look at it waving there. Wave meaning it has peaks, it has troughs, and it moves from peak to trough, back to peak, over a certain distance or at a certain frequency, depending on how you want to think about it. Wavelength, frequency, as we can see, very much related to each other, just inversely. Okay, so here's my good old electromagnetic spectrum there. Let me get that out of the way. So over here on the left, notice high frequency, low wavelength. That means it's very high energy. Gamma rays, X-rays, ultraviolet, all of this, in fact, has enough energy that it's going to get absorbed by our DNA and mutate us. So we don't like this. This isn't great. Try to avoid it if at all possible. Then we hit this narrow band here, which has a nice rainbow color. This is our visual spectrum. So at this wavelength, or at this frequency, we can actually see the light. So somewhere around 400 to 700 nanometers. Anything longer than that, or shorter than that, we don't see, even though it's still photons. When we go longer wavelength, lower frequency, we hit infrared. This would be heat. Microwaves, that's heat that's more specific for water. And then the radio waves, some of which we might even use in our cars if we want to, feel nostalgic or something. A lot less energy in these. We're not so much worried about these or radiating us and mutating us or anything like that. But what we should appreciate is that these are all made of the same thing, photons, vibrating. Just depending on the frequency, our retina may absorb it, and if we do, it's visible light. And if our retina can't absorb it, we don't see it. If a tree falls in the forest, that kind of a thing. Now, that oscillation, as you can see, can vary in terms of wavelength, as we've just gone through. That'll determine the type of light, and within visual light there, it determines the color. The shorter the wavelength, the more blue it is. The longer the wavelength, the redder it is. The brightness of light is determined by the amplitude. Do we have a short peak and short trough, or do we have a bigger peak and trough? The higher the amplitude, the brighter the light. Now, regulation of the pupil is done so by the iris, which is made of two muscles there. We have our sphincter pupillae. When this contracts, it shrinks the pupil because it's a sphincter. And then we have our dilator pupillae. This thing is not a sphincter, no. Instead, when it contracts, notice what it's going to do. It's going to pull that pupil open, so it actually dilates the pupil. So it's a good name. Makes a whole lot of sense. The sphincter pupillae is controlled by both the parasympathetic and sympathetic nervous systems, and only the sympathetic nervous system controls the dilator. So a couple of things are going to regulate the size of our pupil. First of all, light levels. Okay, how can we read this? Low light, bright light. So going from left to right, the light level is getting brighter. What happens to the size of my pupil? That's the height of this. My pupil's getting smaller when it gets brighter. Of course, there's more light. I don't need as big of a hole in my eye. If there's less light, I need a bigger hole to let in that dim light. And you can see we can vary the amount of light coming in by about 5-fold. And this keeps the amount of light on our retina fairly constant. A couple other things that can affect the size of the pupil is, of course, well, how do we feel? Our emotional response. So when we have an emotional response, whether it's good or bad, notice what happens as we go from left to right. Our pupil is going to get a little bit bigger. And object distance. As the object is further away, our pupil gets a little bit bigger. Or put another way, as the object is closer, our pupil gets smaller. And this probably helps a little bit with focusing the light on very near objects. The lens is regulated also by the sympathetic and parasympathetic nervous systems. This has everything to do with that ciliary muscle. So here it is, nice and relaxed. Notice there's a big inner diameter here. Yeah, that's pretty bad. Let's go with that. Yeah, that pops a little more. Okay, cool. The inner diameter, much greater here than down here. So the relaxed ciliary muscle there pulls harder on the lens and makes it flat. That allows us to focus on objects that are further away because the flatter lens doesn't refract the light as much. However, when we look at something that's up close, that light is spreading out more. We need to refract it more. The ciliary muscles contract. That decreases tension on the lens and it widens out. So there's more lens that the light passes through, thus it gets refracted more, and we can focus on closer objects. And this works pretty well as long as our eyes are fairly spherical. If they're a little off, that's okay. We're all snowflakes, no big deal. So if we have farsightedness, it means our eyes are a little short, and so we don't have as much of a distance to refract that light. Oh no. So we can see things that are really far away, but close objects are really hard to see. What do? Use some convex lens, which will start the refraction process before light gets to our eyes. So then we can accommodate that shortened distance for refraction. If we have the opposite problem, where our eyes are a little long, well, we have more space to refract light, so now we're actually focusing it a little too close. This would be nearsightedness, so this works fine enough for near objects, but far away objects, we have a hard time seeing them. So what do? Concave lenses actually spread the light out a little bit more, so that additional refraction that occurs because of our long eyes is totally fine. By spreading the light out, we now focus a little further back, which is exactly where our retina is. Okay, we got a couple reflexes to control our iris and our ciliary bodies. We'll start off with the photopupillary reflex. This one is really commonly used. You shine a light into someone's eye and the pupils constrict. How does this thing work? Just a handful of neurons here, no big deal. Okay, so we shine some light over here. Let there be light. So we'll increase the amount of light here. That's what's going on. We have more light, and what does that do? That stimulates this neuron here that forms the optic nerve. So that's my first neuron over here. Check, it's activated by light. That's going to project on back into the midbrain, and now we're going to activate this second order neuron in the pretectal nucleus. Boop, there we go. That one's active. Now notice what it's doing. It's projecting bilaterally to both sides on this Edinger-Westphal nucleus. So even though we shined a light in this eye, in the, I suppose, right eye, that's going back, and it's going to stimulate both the right and left sides so that both eyes will respond. Those second-order afferents then turn on both sets of the next neuron, the preganglionic parasympathetic neurons in the Edinger-Westphal nucleus. So now we're getting output. So now it's my first efferent, not an afferent, but an efferent. Okay, those project on out. Both were active, so both of my postganglionic neurons are active in the ciliary ganglia. Check those off, and what do? They stimulate the sphincter pupillae there to contract a little bit. Let's see if we can symbolize that. That'll work, okay? And my pupils get smaller. What does that do? What's the purpose of this reflex? Decrease the amount of light that's hitting my eyes. So let's see if we can summarize the photopupillary reflex. Of course. Okay, sorry about that. Got a fat forearm, I guess. So if we increase the amount of light that hits the pupil, that causes, through these four neurons, a decrease in pupil size, which really is a decrease in the amount of light that hits the retina. Sweet. What happens if we decrease the amount of light? I'm sure you're way ahead of me. Well, that, of course, turns off this reflex, so my pupillary sphincter relaxes, and now my pupil dilates. And that, of course, increases the amount of light that hits my pupil. The photopupillary reflex is all about giving me stable light levels. If there's an increase in light, decrease the amount that's coming in. If there's a decrease in light, increase the amount that's coming in. The near response, or the accommodation reflex, deals not with changes in light levels, but changes in where we are focusing. So when my eyes converge, that is, I'm looking toward the middle, if you cross your eyes, you stimulate this reflex. What happens here? So whenever we choose to look at something closer, our eyes come together, and this is controlled by vergence cells. These are cells, obviously, that cause convergence of the eyes. And they're going to do two things. The first thing they're going to do here, not so nifty or anything, this first one is just to activate that oculomotor nerve, and in particular the parts that serve the medial recti in both eyes. That will cause both eyes to come toward the midline. Great. But the reflex part here is what happens next in part 2. The other thing that vergence cells activate are those Edinger-Westphal neurons. Yes, indeedy, they are the exact same ones that constrict the pupils. So we get a little pupillary constriction. It is indeed true. If you cross your eyes, your pupils will constrict just a little bit. The other thing that we're going to hit in this case is the ciliary bodies there. So not only we're going to hit the iris here, we're also focusing on those ciliary muscles, and what's going to happen? They're going to constrict. It's what we call accommodation. So that constricted ciliary muscle causes the lens to thicken up because we have decreased tension on those suspensory ligaments, and now we can focus on things that are close. Why would we want to do that? Because our eyes are converging. So clearly we're looking at something near. And when we do that, we need to focus on close things. That's why we thicken the lens. And constriction of the pupil is supposed to also assist with focusing on very close objects. Those ciliary bodies are, of course, going to be stimulated by the parasympathetic input here. For those of you aficionados who care, these are M3 muscarinic acetylcholine receptors. What really matters? GQ. In other words, it's excitatory. The sympathetic division, as you're probably suspecting, does the opposite, and that's because its receptor has a different G protein, GS, which for smooth muscle inhibits. So this relaxes the ciliary muscle, causing increased tension on those ligaments, thinning of the lens, and thus the ability to focus on things that are further away. And that should make sense. When do we have a sympathetic response? Whenever there's something that we need to worry about, right? Something that requires fight or flight. Okay, this pathway is a little bit different. We're not coming out of the midbrain like we were with parasympathetic stuff, using that oculomotor nerve. Instead, we got to go down to the spinal cord. This is where sympathetic neurons live. So let's say that you've decided maybe there's a predator nearby, we need to be on the lookout. That's going to start up here in the brain, in the hypothalamus. Project on down into the thoracic spine. That's where the first efferent lives here. That then projects on out and activates the second efferent, the postganglionic sympathetic neuron that lives in the superior cervical ganglion. This then crawls its way back up into the head there, following along the carotid arteries, forming the ciliary nerve, and finally we hit our targets. So a much more convoluted path. The parasympathetic path is this. There you go, from midbrain to eyes. And then here's my sympathetic path. Okay, one of those is way more direct than the other. Now, what's the effect here? Well, we raise our eyelid. Again, we're looking for stuff. Okay, so we're going to stimulate the eyelid there. I think that might be Mueller or something like that. Doesn't matter. We raise the eyelid. What else do? We dilate the pupils. We're looking for stuff. We need more light to come in. So we're going to stimulate the dilator and inhibit the sphincter. The other thing we're going to do is flatten that lens by acting on the ciliary muscles, as mentioned previously, allowing us, again, to focus on things far away. We want to detect the threat before it gets to us. That's the whole idea there. The other thing that can turn this on, besides, us imagining there's a threat, you can pinch someone's neck, and that pain will trigger the ciliospinal reflex, not the cilioid spinal, no, ciliospinal, dealing with the ciliary nerve there. And you'll see a little bit of pupil dilation as a result. Now, when these fibers are damaged, typically it's going to be, the postganglionic neuron there, but it could be the pre. Whenever we have damage to this sympathetic input, we get what's called Horner's syndrome. And let's think about what that looks like. First of all, drooping of the eyelid. Hey, that'll happen if we lose this sympathetic input. What else do we see? We see constriction of the pupil. Ooh, that'll happen if we lose this input. The only thing not explained here would be the lack of sweating. And that's another function of the sympathetic input. So we'll have what's called anhidrosis. That's the third hallmark of Horner's syndrome. So two of them are effects on the eyes, and the third would be the lack of sweating. Okay, again, make sure you got something out of this. So scan this or follow the link in the video description and make sure this makes sense. Now, finally, we're going to transduce that light, a photon, into electrochemical signaling. That's going to happen first in photoreceptors, and then we're just going to have a whole bunch of neurotransmitters causing the release of other neurotransmitters. So we're going to go photoreceptor. That's step one. Then bipolar cells, then retinal ganglion cells, and finally, we have our optic nerve. The retinal ganglion cells make up that optic nerve. Okay, so we've now focused our light. We made sure there's enough light coming in and that light has hit the retina. That's where we're at in this story now. Now, the whole point of our optical components is to make sure the light that hits the retina, like we're seeing here, is in focus and that there's enough of it. Not too much, not too little. So on these photoreceptors here, that's what all these little hexagon-looking things are. Those are photoreceptors in the fovea. And notice that some of them are darkened by this E, some of them aren't. And so the shape of what we're seeing creates a shape of activity in the retina. If it's focused, we'll have these nice crisp edges. If it's not, then, well, we won't. Now, another thing that we can take away from this image, take a look at my retina here. See, there's the retina. We don't care about this pigment epithelium right now. The photoreceptors, notice where they are. They're in the back of the retina. So light has to pass through these retinal ganglion cells, these bipolar cells, and then it finally hits a photoreceptor. This isn't a big concern in the foveola, that is the very, very center of your visual field, but everywhere else, we've got all these cells in the way that kind of refract the light and make it a little blurry. This is a backwards way of constructing the retina. Hell, I don't know why, but this is just how it was made, and in evolution, if it works, then it works. This is clearly, though, not intelligently designed. Squids have the retina oriented with the photoreceptors toward the light, which is probably how you would design it. It's how I would design it too. So either there's no intelligent design or squids are the real favored animal out there. Okay, so our retina is backwards. So keep that in mind. This is actually the front of the eye down here, and the back, the sclera is back here. We got our choroid for the blood vessels. In the retina, what are we hitting? This pigment epithelium. The whole point of this is to just make sure that whenever light passes on through, it gets boop, absorbed and it's gone. It doesn't float on through and then hit something shiny and bounce away and scatter and start to stimulate other parts of the retina that it shouldn't. So pigmented epithelium just prevents reflection, so light only gets one chance to stimulate those rods and cones. Okay, then we have our outer nuclear layer. That's right here. So there's 3 layers we're going to go through. First layer being the outer nuclear layer, and that is right here, rods and cones, the photoreceptors. It's on the outside. That's why it's called the outer nuclear layer. Then we have the inner nuclear layer. That's our second layer there. That's right below it. That's the next one. And that's where we have some interneurons. Which ones do we care about? Absolutely the bipolar cells. We have to talk about those. These are the relay between photoreceptor and the retinal ganglion cells. So the photoreceptors pick up the light, tell the bipolar cells about it, and then they tell the retinal ganglion cells. So it's a 3-neuron relay, 2-neuron relay in here. In the internuclear layer, we also have horizontal cells. This is for the lateral inhibition. I'm not going to focus on it too much in this class. I do have other videos on this that you can check out if you really want. And then amacrine cells, which are just way too diverse and complicated for us to spend any time on. So we are going to just brush these under the rug for the purpose of this class. But they do exist, so, they're on the slide. The third layer there is the ganglion cell layer, and that's, of course, on the bottom here. And this contains my retinal ganglion cells, the neurons that project on out and form the optic nerve. So we'll walk through these layers. We'll start with the photoreceptors there, and they come in 2 flavors. There's the rods, which are bigger, and the cones, which are a little smaller. The rods are color insensitive. Because they're larger, they can absorb lower levels of light. So this makes them very useful for dim settings. Cones, a little bit smaller. These do distinguish between different wavelengths of light, so they're color sensitive, but we need a little more light to turn them on. Now, while we call rods and cones the photoreceptors, the actual receptors called a photopigment. So this will be the protein there, the opsin, that actually detects the light. And opsins only absorb light because they hold on to a retinol. So this is the pigment Retinol is the pigment, opsin is the receptor. Retinol absorbs light at around 370 nanometers on its own, but when it's bound to an opsin, that changes that sensitivity into somewhere in my visual spectrum. It depends on the opsin. That opsin is also responsible for actually creating the intracellular response because it's the G protein coupled receptor. Now in rods, they express an opsin called rhodopsin, which is actually kind of somewhere in between all these. It's kind of a broad wavelength opsin, and there's only one of them, so it's not color sensitive. Now, cones express color sensitive opsins called photopsins, not rhodopsin, and there are three types: long, medium, and short, LMS. And this deals with the wavelength of light. L-opsins, long wavelengths. We call those the red opsin. You'll notice its peak is right here, more in the chartreuse part of the spectrum, but okay, we'll call it red, even though that's not the peak. M's, we're going to call green opsins, even though it's maybe more of a sea foam, that's okay. S, short wavelength. That's going to be down here in the violets. We're going to call them the blues. So here we have it. RGB, red, green, blue, long, medium, short. And this is the basis of how we pick up colors. Now you'll probably notice if you've ever played with any software like Photoshop, right? They have RGB sliders that let you adjust the color. So our retina does the same thing. We've got our RGB detection right there, and that creates my color opponency that we'll look at in a little bit. Red, green, and blue, yellow. These photopigments are stacked into these photopigment discs. So we got a whole bunch of discs stacked up in this rod. That's what the top of it is over here. So a bunch of these discs continually created and shed, and within that disc, it's just lined with these photopigments, which again is the opsin and retinol combination. These discs eventually get worn out and we shed the old ones on top. This thing can, okay, there we go. This thing can pop off here and then we'll recycle it, right? We'll phagocytose it and recycle what we can. Now the retinol within the opsin exists in two states. There's the cis retinol, and this is pigmented. This can absorb light. And what we're talking about here, let me go ahead and mark it, is this. That angle right there at that double bond, that can exist either as cis retinol or trans retinol. When it's bent like this, forming cis retinol, this can absorb photons and thus it is pigmented. After it absorbs a photon, it then changes to trans retinol and is no longer pigmented, so it's what we call bleached. Cis retinol inhibits opsin. So opsin is not active when it has cis retinol. Only when it's holding on to trans retinol is it active. Okay, why does that matter? Well, this is the basis of large adaptations in light levels. Okay, let's review this illustration here first of all. So here we are, we got opsin, cis retinol, it is not active. If there is light, cis retinol absorbs the photon and now it's trans retinol. So this opsin, while it's holding on to it, let's say green here, green is go. This is active. It does stuff. It creates some signaling cascade, and we now believe, hey, there is light. But this interaction is not stable, so that means that this trans retinol quickly dissociates from opsin. So this opsin down here is no longer active. So there's this brief window of time when opsin is actually stimulating its signaling cascade, and that's right after that cis retinol absorbs a photon, it turns into trans retinol. After it dissociates, no more opsin activity. In order for this opsin to become active again, trans retinol has to be turned back into cisretinol and then rebind the opsin, but that only happens in dark conditions. What's the beauty of this system? So if it's really dark, then we have a whole lot of cisretinol. So let's say you're sitting in a movie theater. It's dark. You're watching the movie, but as you're sitting in the dark, what's going on here is we're increasing the amount of cisretinol that's in the retina. In other words, your retina becomes more pigmented. That means it can more easily absorb low levels of light. So when you're in the dark, you're able to sense low levels of light. Let's say you then walk out of that movie theater, you go out into the world, it's noontime or something, it's really bright, and it's so bright that it hurts your eyes. It's not that pupillary light reflex that's going to help you. No, it's the bleaching of your retina. So you have a really pigmented retina right now. What's going to happen when it's light out? So if it's light, well, then it's not dark anymore. And we're getting rid of that cisretinol, and what we're building up is a bunch of transretinol. So our retina becomes bleached, and that means it absorbs less light. That's totally fine, though. We're in bright conditions. There's plenty of light. We don't need to be as sensitive to low levels of light. And so after a while, after we bleach our retina, we're okay. We can see again, and it doesn't hurt. If we go back into that dark movie theater, we can't see for a while, we're totally blind. We wait a little bit while we accumulate more cisretinol, and hey, now we can see again because we have a very much pigmented retina. So this bleaching is going on all the time, and this, of course, along with your pupillary light reflex, tries to maintain a relatively stable level of signaling in the retina. Okay, so now at this point, we've absorbed a photon. We get that good old transretinol activating the opsin. What does the opsin do? So opsin's a G protein coupled receptor, and the G protein here is transducin. That's the one we're dealing with here. What transducin does when it's active, so boop, 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 go over here to step B, now it's active, it turns on a phosphodiesterase. As the name suggests, this breaks phosphodiester bonds, so that's going to destroy cyclic GMP, turning it into GMP. Just regular old GMP, no cyclic bond there. Well, who cares about that? Cyclic nucleotide gated cation channels do. Okay, so let's think about what happens. Let's, what's a good color? Maybe a purple. Okay, so we have more light. We absorb that. That turns on our transducin here via, of course, the opsin. So the opsin's now active. Let's give it some action lines. And now we have activated our transducin. Phosphodiesterase gets activated. Oh boy, there's more activity here, however you want to think about it. We're destroying cyclic GMP. Well, that used to turn on this ion channel, so now this thing is less active. So here's what's weird. That means that light, through all this mess on the slide, inhibits photoreceptors. You probably would imagine that light would turn on a photoreceptor. No, it turns it off. We decrease cyclic GMP. That decreases our permeability, that's the P, to sodium, so we're not depolarized. Instead, we hyperpolarize. We're less active. We don't spit out as much glutamate anymore. That's weird. We're going to return to that, okay? So those photoreceptors, whether they're rods or cones, depends on where they live. They're not equally distributed. So in the fovea, remember the center of my visual field, that's the 0 degrees here. There's my fovea. What are we seeing? Whole bunch of cones right in the fovea, not so much away, nothing in the blind spot, of course. As we move to the periphery, very few cones. Rods are found in the periphery. Okay. Another difference, cones have about 1-to-1 mapping to retinal ganglion cells, whereas rods about 15 or 30 to 1. So what this means is that in the fovea, where we're full of cones, we have very good visual acuity, we can distinguish colors, but we need bright light for that to work. In the periphery, notice, mostly rods with a whole lot of coupling. That means we don't have great visual acuity, but we can detect low levels of light, and we're very sensitive to movement. This thus gives us two systems, two visual systems. There's the photopic system, meaning a lot of light, that's all that means, and scotopic, the low light system. So in the periphery, remember it's about 15 or 30 rods funneling down via bipolar cells into a single retinal ganglion cell. Notice this is the receptive field size for this retinal ganglion cell, about 1 square millimeter. Doesn't sound like a whole lot, but compare that to the retinal ganglion cells over here that have that nice 1-to-1 coupling. Ooh, they're going to be responsive to about 2 square microns, so about 50,000 times smaller receptive field. 500,000 times smaller receptive field than over here in the periphery. So we have much better vision in the fovea because of that 1-to-1 coupling between cones and retinal ganglion cells. Now, of course, the receptive field actually includes the neighbors, so it's not quite 2 square microns, but you get the idea. Much smaller receptive fields in our high light photopic system. So that covers our photoreceptors, finally. So remember, when there is light, what happens? We turn off the photoreceptor, right? It stops spitting out glutamate. Now we move on to the next cell, that's the bipolar cell, and there are two types. There's the off bipolar cells. These are only used with cones. These are normally excited by glutamate. So think about what happens. We go over here to the light condition. The photoreceptor is no longer spitting out glutamate. So that means that when there is light, the off bipolar cell is turned off. So the name tells you the effect of light on the bipolar cell. Light turns it off because glutamate turns it on, and light turns off glutamate release. I know I didn't make this up, so don't blame me. But you do need to say this many times for it to make sense. The on bipolar cell, again, this tells us what light is going to do to it. So light will turn it on. How does that work? Well, remember, when there is light, the rod over here is no longer active, so it's not spitting out glutamate. That glutamate is inhibitory to an on bipolar cell. So we're not inhibiting the bipolar cell. Therefore, it is active, and so it spits out glutamate onto the retinal ganglion cell, stimulating it to start firing a bunch of action potentials. On bipolar cells are used with both rods and cones. We're going to come back to our off bipolar cells whenever we get into color opponency. So, very simple 3-cell relay. We got our rod here, then we get to the first neuron, the bipolar cell, and then the retinal ganglion cell. Now, sure, bipolar cells talk to other things besides retinal ganglion cells, but we're not going to sweat that for this class. The retinal ganglion cell then forms the optic nerve. And the retinal ganglion cells that contribute to our vision are always stimulated by bipolar cells. And because of horizontal cells, they have these center surround receptive fields. In other words, if I shine light in the center, I get one response. In this case, stimulation. But if I shine it in the surround or in the periphery, I get the opposite response. This is super important for edge detection. I promise. I'm not going to harp on it in this class, but I do have a video explaining it, which I will put in the description. It's probably not worth watching, truth be told. But, for the sake of completeness, I will put it there. I don't encourage you to watch it, though. Now, there are some retinal ganglion cells that directly detect light. They actually have their own opsin, melanopsin, that picks up blue light. And these are those retinal ganglion cells that create the reflexes, the light reflex that we went through just a bit ago. And these also entrain our circadian rhythms. So when it's bright, we want to feel awake. When it's dark, they make us feel a little more tired by communicating with the hypothalamus. More on that later in this class. So how do we create color vision, right? Rods only have that one opsin. They're not distinguishing wavelengths. They don't care. They're just trying to say, is there any light at all in the periphery? In the fovea, we have our blue, green, and red cones, and they do distinguish different wavelengths of light. Yes, there's a lot of overlap, but they have their own excitability curves here. So we can compare which one's the most active. If I see a whole lot of blue cone activity and not a lot of green and red, it's probably blue. If I see a lot of red cone and not a lot of green and blue, it's probably red. So this is explaining what we call the color opponent processing. So we got our blue, green, and red opsins, SM and L, and these then converge onto color opponent cells in the retina, in the thalamus, lateral geniculate nucleus, and the primary visual cortex. We have color opponent cells. What do we mean by color opponent? So there are these two pairs, and they oppose one another. The example I have here are a couple of red-green color opponent neurons. Red on, green off. If there is red light, I'm active. If there's green light, I'm inactive. I'll call this Ron-Goff. I have another color opponent. Green on, red off. Gon-Roth. So based on the activity of these two neurons, I can tell whether it's green, red, or yellow. Let's take a look here. So let's say that there's green light. Now remember, that turns off the M cone, which turns on its on bipolar cell. And thus we stimulate Gon-Roth a little bit over here. What else is going on? It's also getting input from this off bipolar cell that listens to the L cone. Hey, there's no red light. So the L cone isn't hyperpolarized, so it keeps spitting out glutamate onto this off bipolar cell. And so this Gon-Roth over here is now doubly excited. It must be green. What if it's red? The opposite happens. We inhibit the L cone, not the M cone. So the on bipolar cell is active downstream of the L cone, and the off bipolar cell is active downstream of the M cone. Now it's Ron-Goff over here who's doubly active. Gon-Roth is asleep. What if it's yellow? I promise, yellow is a combination of green and red. I promise. Trust me on it, or go play with some RGB sliders. So both the M and L cone absorb yellow light. So both of their on bipolar cells are on, and their off bipolar cells are off. This means I get a little bit of excitation over here on Gon-Roth and a little bit of excitation over here on Ron-Goff. So if it's red, only Ron-Goff is active. If it's green, only Gon-Roth is active. And if it's yellow, both of them are kinda active. And there's my red-green color opponency. The same system is there for blue, yellow, except in this case, we need the S cones. So there might be a blue on, yellow off neuron, Bon-Yoff. That's stimulated by the on bipolar cell downstream of S cones and by the off bipolar cell downstream of L and M cones. Because remember, green and red makes yellow. We might have a yellow on neuron. Well, that would be Jan-Boff. On bipolar cells downstream of M and L cones turn it on, as does the off bipolar cell downstream of S cones. Draw it out. It's a ton of fun, I promise. So that's when color vision works. What about when it doesn't? So with color blindness, most commonly what we're dealing with is we're missing an opsin, or it's mutated, so it doesn't work. Most commonly, it's an M or L opsin giving us red or green color blindness. And that's most common because, well, these genes are very similar to one another, and they're probably derived from the same gene that then mutated over time following a duplication. So sometimes we get crossing over occurring, and we delete the gene, and it's on the X chromosome. And that makes it very common in males compared to females, because males, of course, only have one copy of the X chromosome. And we can detect this color blindness by doing something like an Ishihara test. So hopefully you can see 16 here. Everyone should be able to see 16. If you have red-green colorblind, you're not seeing the 3 or the 10. And unless you have blue color blindness, you're seeing the 42 pretty well. You might just see a 4 here and some kind of gibberish over here if you're blue colorblind. Let's see what that looks like. Let me get this out of the way. So if we have red or green color blindness, here's how it looks. Maybe we can kind of see something going on over here with the I think the 3 was there. Yeah, there we go. Maybe there's something, but I wouldn't pick out a 3. I don't see a damn thing down here with the 10. The 16 and the 42 still look pretty good. If we have blue color blindness, well, then, of course, we can see the 16. Everyone can see the 16. Now the 3 and the 10 are back. But the 42 is a little weird. The 4, I think, still is visible to me. Without knowing it, I wouldn't be able to say this is a 2. It might be an 8, maybe kind of a 2. But notice it doesn't look anywhere near as good as, back here or here. So once we stimulate those retinal ganglion cells, right, let's recall what's my pathway here. So again, 1, 2, 3 neurons, like a whole lot of signaling pathways. The first neuron is the bipolar cell. That activates the retinal ganglion cell. That's my secondary afferent. That forms the optic nerve. Just like many other signaling pathways, the secondary afferent is the one that can cross the midline. So a little bit of jargon here. The optic nerve comes off of the retina, and these two optic nerves converge at the optic chiasm. And here, some fibers will cross over. Now, it's not dependent on the eye. No. It really is all about the visual field. So vision is contralateral, meaning the left visual field, regardless of which eye it hits, goes to the right side of the brain. So my right eye that sees the left visual field stays on the same side. My left eye, on the other hand, that hits the left visual field must cross over. So this is what's called the hemidecusation, meaning some of the fibers cross, but not all. Here I'm at my tertiary afferent, that be in the thalamus, in particular the lateral geniculate nucleus of the thalamus. Another bit of jargon here, the optic radiations are the axons that go from the LGN to the primary visual cortex. And then we start to make sense of what in the heck am I looking at? And there are two parallel processes going on. First, we want to figure out what is it, and that's the ventral stream. So in the primary visual cortex, lots of neurons figuring out very bitsy details. What's the angle of the line? Does it stop? Does it not? What are the lines and edges, in other words? And then based on my lines and edges, we go to the next stop. We go from V1, primary visual cortex, to now this V2, the next step in my visual cortex. And hey, I've got 4 lines here that stop around there and make those angles. Hey, that's a square, so I can figure out what shapes there are. Okay, go on down the line to the next stop, and we turn shapes into objects. So notice what we're doing. We're stepwise building what the heck am I looking at? And we continue along into our temporal lobes, and then there will be there are places in the temporal lobes that are dedicated to very specific bits of information, like, am I looking at a face? Okay, whose face is it? It's not a face. Is it a guitar? Is it a watermelon? I don't know. That's what's going to get figured out over here. And what we're going to do, notice we're in the temporal lobes, we are very close to memory and language regions. Okay, places like the hippocampus, also emotional places like the amygdala. So how do we feel about it? Do we have a history with this thing? And so following the ventral pathway, we figure out, what am I looking at? At the same time, there's a dorsal stream here that's on this slide. Okay, the dorsal stream, now this is the visual cortex, so the brain is flipped around. Now we're looking to the right. The dorsal stream goes more along the outside and then up into the parietal lobes. Now you're probably noticing that if you look up here, this image looks like it's moving. Until you look at it, then as soon as you look at it, you realize that's not moving at all. Why is it moving? It's constructed in a way that stimulates these neurons right here in, I think, what's called V5. When these neurons are active, we perceive motion. Whether things are moving or not, doesn't matter. The angles of the lines and the contrast there is created to stimulate those neurons, and so it looks like that's moving. It's totally not, but that's part of the where pathway. We want to know if a thing's moving, and then where is it? Now notice where we're going. Right here. This, by the way, let's go ahead and label this. This is my primary somatosensory cortex. In other words, my body map. That's this strip right here. So this is where I am. Here's me. Here's my visual field. Right there in between, we're going to average those functions. Where is that thing relative to me? That's what matters. If I'm looking for my car keys, I don't really need to know their GPS coordinates or anything. I just want to know, are they in front of me? Are they to the right a little bit so I can go and reach for them? Now, speaking of reaching, we also continue along here to the motor cortex. Oh, what's the point of that? Notice we're hitting kind of like handy regions. This is handy because it helps visually guide reaching tasks. So I figure out where it is, and then I can reach for an object if I so desire. So 2 pathways: ventral, what is it? Dorsal, where is it? And that's pretty much vision. Now just a few visual illusions, because these are always fun. Notice that our visual perceptions are relative. We're not trying to say it is exactly this gray or this white or this red or whatever. We form our perceptions relative to the things around it. So squares A and B, you probably already can guess that they are the same color, but they should not look the same to you. If they do, please go see a neurologist. Something is wrong. A should look darker than B, because look at A. This is clearly a checkerboard pattern, and it's next to the light square, so this has to be the dark one, and B is surrounded by the dark square, so this has to be the light one. But if we move a little bit of A over to B, look, it disappears. It's the same color. I promise. Let's expand that, and look, these are the same, but they don't look the same. I know they're the same, but there's this little nagging homunculus in my head saying, no, they're not the same. A is clearly darker than B. Yeah, they're the same. But that's not the purpose of our visual system, to tell me exactly how gray it is. It's for me to figure out, how does it look relative to the rest of the world? Okay, we have a look here. Clearly we have like 1, 2, 3, I don't know, kind of purplish, pinkish dots, and then like 1, 2, 3 kind of reddish dots, something like that. Of course, that's not what this is. They're all the same pink dot. It's just we have these green lines and these blue lines that are covering it, and that makes the pink look a little redder here and a little more purple up there. Okay, one last one here. This is a fun one for 4th of July coming up. So stare at that X. Please don't move your eyes at all. Don't blink if you can help it. No, the video's not broken. It's still there. Continue to look at the X. Don't move. Don't blink. Ah, how nice was that? So that's a nice little patriotic way for us to end in this video here. This is showing us how the visual system accommodates. So what did we do? We showed the opposite color. We showed the color opponent for the American flag there. And what we did was tire out those colors. All right, so if we go back, yellow, what's the opponent of yellow? Blue. Right, what's the opponent of black? White. What's the opponent of this particular shade of blue? It's actually red in that case. So by tiring out your yellow neurons, when this disappears, those yellow neurons are really tired. So they rest at a much lower rate than they would normally. But the blue neurons right here haven't been tired out. So we see more activity in blue neurons than yellow. So hey, must be blue. No, not at all. That's not what's going on. Okay, and I've totally screwed this up. But anyway, here, let's just go around this. There we go. All right. I don't want to wait the 20 seconds, and neither do you. So again, you can scan this. You can follow the link. Make sure you're getting something out of this. And if there's anything that's unclear, let me know. Email is great. I guess you can comment on this video. That's fair game too. Just, be nice. All right. That'll about do it. I'll see you later.